Simple Mendelian inheritance means that we have one dominant allele that completely masks our recessive allele. The truth is most genes are not inherited using simple Mendelian inheritance. So let's talk about something that's a little bit more complex, and that is incomplete dominance. Flower color in snapdragons is controlled by two alleles that are incompletely dominant. Cross a red flowered plant with a white flowered plant. Predict the genotype ratios of the F1 and F2 generations. First, let's talk about notation. We are going to have, in this example, one dominant allele and one recessive allele. And in this case, our dominant allele produces a pigment for red. The recessive allele has a mutation in it that doesn't produce any pigment. So the phenotype associated with this allele is white. So far this looks pretty standard, but where we see a difference is when we look at the genotypes and the associated phenotypes. So let's spell it out. If we have one individual that has two dominant alleles, that means they have two of these genes that code for the red pigment. So the phenotype, or the appearance of those flowers, is going to be a nice deep red color. On the other hand, if we have r, homozygous recessive, this individual has two mutated genes and does not have the ability to produce red pigment, leaving those flowers white. With our heterozygous individual, big R, little r, this plant has one dominant allele, so it will produce some red pigment, but not as much as the flower with the big R, big R. So the resulting phenotype here is pink, which is intermediate between the two homozygous phenotypes. So to compare that to Mendel's simple dominance, if we had big R, that coded for red, big R, little r also coded for red. So the difference with incomplete dominance comes mainly when we look at the heterozygote. Now that our notation is defined, let's go back to our sample problem. Cross a red flowered plant with a white flowered plant. So our parents, we have a red flowered plant, and there's only one option. That genotype must be big R, and our white flowered plant will be little r. When we make the Punnett square, well, before we can make the Punnett square, the gametes produced by each plant, there's only one option for both plants. This plant that has big R only has that one type of allele. So every single gamete produced by this plant will carry a big R. Using that same logic, we know that every gamete produced by this little r plant will carry little r. So when we make our Punnett square, it's going to be really small. One parent can only produce big R gametes, the other parent can only produce little r gametes, and our Punnett square is only one box. There's only one possibility for offspring, and that is that they will all carry a big R and a little r. So remember that the offspring produced by this first set of parents are called the F1. So our genotype ratio for F1, right, yep, we're asked for genotypes, not phenotypes. So that is 100% big R, little r. We also need to figure out the ratio of the F2 generation. And to figure out the F2, we just take two F1s and cross them. So in this case, our parents will be big R, little r, crossed with another big R, little r. This first parent can produce gametes that carry the big R and gametes that carry the little r. Let's make that clear. These are the gametes produced. Same thing with this parent. Half of its gametes will contain the big R, the other half will contain the little r. Our Punnett square will need to be 4 boxes. And we can do the cross to figure out the genotypes of these potential offspring. Our genotypic ratio, remember, is just a list of the different possible genotypes along with how many of each there are. So the first genotype I see is big R, and 1 out of 4 of these potential offspring carry that genotype. The next possibility is a big R, little r, and there are 2 of those. So 2 out of 4, but we want to reduce that to 1/2 big R, little r. And the last possibility, little r, and 1 out of 4 carries that genotype. Remember that you can also use percentages or whole numbers for your genotypic ratio as well.